Our speaker today is Anu. Anu is a master's graduate from the Computational Science / Data Science program here at SDSU. She studied under Dr. Naveed Vaidya from the Mathematics Department, and her master's research focused on mathematical disease modeling for HIV particles. So thank you, Anu, for being our speaker, and you can take it away.
Let me share my screen. Can you see the screen? Okay, everything's good. Okay. Let me make this. So, hello everyone. I am Anu. I just graduated a little over a month ago from SDSU, and I'm currently in New Jersey. Welcome to my presentation on modeling spatiotemporal distribution of HIV particles on cervicovaginal mucus, or the CVM. This project was a part of my master's final project, and I worked with Professor Naveed Vaidya from the math department. I worked on this for, I think, a year and 9 months. So yeah, long project. Okay, so starting with the outline, I will begin with some introduction, give some biology context to what is happening here, and then I will move on to some objectives and some justifications. Then I will talk about the model development and results, and end my presentation with conclusions. Starting with the introduction, HIV epidemics remain devastating around the world, with an estimated 38 million people currently living with the virus worldwide, 2.1 million people being infected yearly, and about 2 million deaths annually. So HIV is transmitted through certain bodily fluids, such as blood, semen, vaginal fluids, etc. And it is a virus that attacks the immune system, specifically the CD4 or T cells, which are essentially our white blood cells that help body fight off infection. So HIV is a chronic condition. So if it is left untreated, it can lead to acquired immunodeficiency syndrome, which is popularly known as AIDS. And so a critical factor in HIV transmission is the presence of virus in a genital secretion, such as the CVM. So for the virus to essentially enter the female body and reach its target, it must first cross the CVM, which is this thick acidic fluid, which is produced by the cervix. And this acidity of the CVM may hinder the motion of HIV and other viruses and make it more difficult for them to survive and replicate. The spatiotemporal distribution of HIV particles on the CVM can affect the likelihood of transmission and may be influenced by factors such as pH and presence of other microbes in the region. So despite the importance of this issue, very little is known about the dynamics of HIV particle distribution on CVM and how it relates to transmission risk. And this is what we wanted to answer through this project. So in the absence of HIV vaccine strategies delivering microbicides that destroy HIV or prevent its entry into the vaginal mucus have been considered for a potential HIV prevention. Because we don't know so much to address the knowledge gap here, we have developed a mathematical model to predict the distribution of HIV particles on CVM and understand the role of mucus in the transmission. So the model that we developed here will incorporate pH-dependent hindrance as well as diffusion to accurately capture the motion of HIV particles on the CVM. And the results of this study will inform the development of prevention strategies and continue to contribute to a better understanding of HIV transmission. Previous experimental studies have indicated that motion depends on acidity, mainly due to adherence to mucus site. So in the figure you see here, it is a 20-second trajectory of HIV particles in the acidic and neutral CVM. So here we see in the acidic CVM, the movement of the particles is very restricted. In about 20 seconds, it's more or less like the particle is in the same location. But whereas in the neutral state, we see that the movement, the particle is all over the place and moving around. So we are sort of seeing this, that when the pH is acidic, since the particle is not moving as much, we want to make sure that we can destroy it when it is in place. So as I mentioned earlier, for the virus to enter the female body, it must first cross the acidic CVM that protects the area from infections by creating an inhospitable environment for pathogens to survive and replicate. So, so since the motion of the virus in the acidic CVM is hindered, accurate modeling of HIV transmission is super critical for understanding how the virus spreads and for developing effective prevention strategies. So essentially, once the virus lands on the surface here, it has to pass through all of these epithelial cells, and then we have all the mucus in between, and then reach its target, which is the white blood cells. So when the HIV is in between in this region in the mucus, it doesn't really affect the person. So the whole idea is to make sure that the virus just stays in the mucus as long as we can, and then try to make sure that all the virus is destroyed in that region before it reaches its target. So the objectives of my study here are essentially to develop a precise mathematical model to investigate the complex factors that impact the distribution of HIV particles on the CVM under various different conditions and quantify the risk of HIV transmission. And secondly, it is to show that proper implementation of nanoparticle-based therapy can significantly control the virus entry through the CVM, thereby avoiding establishment of HIV infection. Now moving on to the justification of this project. As we all know, mathematical modeling is a very powerful tool that can help to understand the transmission and the spread of HIV, as well as the effectiveness of prevention and treatment interventions. So by creating these mathematical models of HIV transmission, we can sort of simulate the spread of the virus under different scenarios and make predictions about the potential outcomes of different interventions. This study can contribute to filling some of these knowledge gaps by investigating the role of mucus acidity in the HIV transmission. So the main reasons that we are doing this project is, or we did this project, is the absence of a cure for HIV and limited success of traditional prevention methods, and which highlights the urgent need for new prevention strategies. And secondly, this research aims to develop a novel approach to prevent HIV transmission and help curb the global epidemic. And finally, this research could lead to a better understanding of the interactions between HIV particles and the mucus, which may have broader implications for preventing other sexually transmitted infections. Moving on to the more fun part of the project, which is the model development. So the movement of the virus, as I mentioned earlier, in CVM is freely diffused in neutral pH, and the movement is hindered because of mucoadhesion due to acidic pH. So we model the spatiotemporal dynamics of virus concentration using two model components, diffusion and hindrance. So here basically V is the concentration of the virus particles at location X time T. So we are modeling the difference in the number of virus particles at each location with respect to time. And here we have the two components, as I mentioned earlier. We have the diffusion, which sort of is just like the basic heat equation, and the hindrance is which captures the pH and how the movement is hindered. Yeah. Okay. So diffusion is modeled using Fick's law, where diffusion is dV delta V. Here dV is the diffusion coefficient and delta is the Laplacian operator. And a term representing hindrance will be derived from the biochemical kinetics that you can see on the screen here. V is the number of virus particles, and M is the mucus. So here we have an attachment rate Kb and a detachment rate Kd. So with some attachment rate Kb, we get Vm, which is the mucus with the virus. And then we have another rate here, Kc, which is the CVM bound HIV loss from CVM. And we get V bar and mucus. So V bar is basically the virus that got stuck, but then got unstuck as time progressed, and then M is mucus here again. Okay. So essentially now we convert this biochemical kinetics into a set of differential equations. Anything that goes to the other side gets a negative sign. The one that's getting added gets a positive sign. The one thing to take away from here is that the change in the number of virus particles with respect to time is 0, and the change in the number of mucus sites with respect to time is also 0. So that is why we have the d of total m by dt is 0 and d of total v by dt equals 0. So essentially, we are saying that the number of mucus sites throughout the process is constant, and the number of virus particles is also constant. And throughout the project, we depicted it by M naught and V naught as the constant number of mucus sites and the constant number of virus particles. One more thing to add is we put 10 to the power 5 as our constant number of virus particles. So almost any results you will see is when the initial number of virus that entered the region is 10 to the power 5 virus particles. Okay. So in addition to all of the equations before and everything, we have also the quasi-steady state hypothesis, which says that dVm by dt equals 0. This is essentially saying that the virus is moving and so is the mucus. So essentially for the virus, we are sort of saying that the mucus is not moving according to the virus. It's just like we are moving and the earth is moving, but we don't necessarily feel the earth moving. Same thing here. So we say dVm by dt equals 0, and then combining all the equations that we have, we get our hindrance as negative mv divided by phi plus v. So here, small m is kc times m naught, and phi here is, the kc times m naught is the maximum number of virus loss, and phi here is kd plus kc by kb. So now that we have our hindrance term, which is negative mv divided by phi plus v, and the diffusion part here, which is dv delta v, we just put them both in here, and we get our final model for spatial and temporal dynamics of free virus in the mucus. So as revealed in experiments, the hindrance motion of HIV particles in acidic CVM is due to mucoaddition, and the pH affected due to acidity change most likely affects the detachment and attachment rates, which are Kb and Kd. And we formulate the value of phi using, here A essentially is the pH value, and gamma here is the exponential decay of phi per unit increase in the pH value. So this is how we are sort of modeling how the phi value is related to the pH. Now, moving on to some results here. First is the transit time. So considering the 1D case, we have 0 to L, where L, I think we've considered 10 micrometers. Here, we have just our equation that I discussed earlier, the boundary conditions. V of 0 is V naught. Here, V naught is again 10 to the power 5 virus particles which are entering into the region. V of L equals 0. And here, in this case, the acidity of the CVM is 7. Sorry, the pH of the CVM is 7, which is neutral state, which essentially means that phi value is going to infinity, which makes this whole term go to 0. And we get our transit time tau naught equals L squared by 2 dV. We do a similar analysis for pH equals 4. We get the phi value equals 0, which essentially means that all of this term just goes to m. And solving this equation, similar to how we did the last one, we get tau m value. And now we use both those equations to get the transit time for both the cases. So for the base case computation where pH equals 7, which is neutral, and our length is 10 micrometers, we have our diffusion coefficient as 0.25 micrometers squared per second. We get the transit time as 3.3 minutes. And similarly, using effective diffusion coefficient 10 to the power negative 3. So this is when we have our pH equals 4, which is the acidic state. And same length again is 10 micrometers. We get the transit time as 13.9 hours. So here we can clearly see that we have all of this extra, almost 13.8 hours to intervene and make sure that the virus doesn't reach its target. Okay. Next here we have hindrance. So from the previous equations, this one, we can get the value of m, which once you have the 13.9 hours and 3.3 minutes, which we got the values earlier for the transit time for both the cases, we get the value of m as 498.6. And we calculate the phi using this equation. Here, we can use any equation here. Essentially, we specifically use an exponential equation just because it's easier to calculate. The only thing we wanted to make sure that was when pH was going to 4, more acidic, the phi value goes to 0. And when pH is going to 7, the phi value goes to infinity. Okay. Two pictures here. The one on the left is mucoadhesion rates for different pH levels. So you can see that more acidic, that is more closer to 4, we have higher mucoadhesion rates, and closer to 7, more neutral state, we have lower mucoadhesion rates. And here in this figure, we see the number of particles that cross within 5 minutes. So when pH equals 4, 0 particles cross the region, but whereas when we have 7 as the pH, more than 100 particles cross. Here, by crossing mucus essentially means reaching the target. And this slide here, this observation has very important implications for the development of preventive therapies against HIV transmission. Basically, it indicates that interventions targeting the modulation of pH in the CVM environment could potentially be used as a strategy to reduce HIV adhesion to the mucus layer and inhibit virus transport. So, for example, the use of pH modulating agents such as microbicides or topical gels that sort of can create this more acidic environment in the vagina may help to enhance the mucoadhesion of virus particles and sort of preventing the further movement and reducing the risk of infection. Okay, some more dynamics here. We see two animations. The one on the left is when pH equals 7, which is neutral state, and the one on the right is when pH equals 4, which is the acidic state. So we can see the one on the left, it takes about 200 seconds for the first particle to reach the target, which is about 3.3 minutes. And here the first particle reaches the target in about 13.9 hours. So you must be thinking these numbers are getting repeated so many times, the 3.3 and 13.9. So the 3.3 and 13.9, the numbers that we get from this slide are from the equation that we are solving. The ones that were previously mentioned are the results, the experimental results. So we sort of can see that this model is accurately representing how much time it takes for the virus in the most ideal situation where you have uniform distribution and the time that it takes for it to reach the target. Okay. So here we did uniform distribution, but in reality, that's not the case. So we wanted to see what happens when we use different distributions like normal distribution. That is what you can see in the screen here. In all the 8 cases here, the mean is 3. Essentially, what I wanted to show here is first of all these 4 are when pH equals 7. So all these 4 images are when we have neutral state, and these 4 images here are when we have the acidic. So here at 80 seconds, just use having different standard deviations. So here the standard deviation is 1, and here the standard deviation is 2. Though it doesn't really strike out as much that 0.78 particles reach here and 0.69, but you can see that the distribution is completely different. And this is something that we need to know because we sort of want to understand what is the best case and worst case scenario on how fast a virus can reach the target and how slow, and just understanding how the medication needs to be applied and just understand how at each point in the region, how the virus concentration is. So that is the whole point of us doing different means and different standard deviations to understand how the virus is just behaving in the region. Okay, this one's just sort of a extension of what you've seen earlier. In this figure here, standard deviation is put constant, which is 1.5, and the mean is changing. In this figure here, we wanted to see how many particles reach one single point, and that point was 5 micrometer, 5 micrometer, which is basically the center of our graph. So it is sort of obvious to think that if the mean is towards the left or towards the right, it would take longer for the particles to reach the middle point. So that's why we see that when the mean is going away from 5 on both sides, the number of particles that reach the target at a particular time point is less. Same thing here with standard deviation. So here we have mean constant at 5 micrometers, and the standard deviation is between 0.5 and 2.5. So essentially, the more the spread, it takes longer for the virus to reach, but then as the spread is just almost concentrated at like a few points somewhere in between or anywhere anywhere on the first row there, you would have more number of particles reaching the target. Okay. So like any scientific study, this project has several assumptions and limitations that should be acknowledged. And yeah, like, firstly, the study only considers the distribution of HIV particles on the CVM and does not account for other factors that may affect the increase in risk of transmission, such as presence of other sexually transmitted infections or the use of hormonal contraceptives. And one more thing to also note is that because there's also involvement of semen here, which is usually neutral in state, it can also reduce the acidity of the region. So which necessarily means that what it essentially means is that the virus, the mucus pH is not as acidic as we think it is. And the model also assumes a constant rate of virus production by infected cells, which may not be accurate. And in reality, the rate of HIV production can vary between individuals and over time. And we are also assuming just one single type of HIV particles and does not account for potential effects of viral diversity of on transmission dynamics. Okay. This slide covers the software methodology. So in this project, I have simulated both 1D and 2D diffusion equations using finite differencing methods with implicit time stepping. And all of the simulations have been done using MATLAB. And this is all the process of how I did it, which was just defining the parameters, pre-allocating field variables, specifying boundary conditions, initial conditions, and calculating coefficient matrices. In the end, as conclusion, our software tool has successfully analyzed the spatiotemporal distribution of HIV particles on CVM, providing a much more accurate understanding of virus transport through the mucus layer. Secondly, our study highlights the potential of mathematical modeling and computational tools to aid the development of effective HIV prevention strategies. And finally, future research will be focusing more on incorporating HIV filled with drugs into our model to further investigate the impact of preventive therapy on the distribution of the virus. So essentially, overall, this study demonstrates the potential of mathematical modeling and computational tools, and we hope that all these findings will contribute to the ongoing efforts to combat the global HIV epidemic. Just a little bit of what is going to be done in the future with nanoparticle-based therapy. As you can see, just this part is what we already have from our equation, but now we have two PDEs. We have a system here, one representing the movement of the virus and the other representing the movement of the medication, the nanoparticles. So we have two different diffusion coefficients, and here this term is how the virus is interacting with the nanoparticles. In this figure here, you can see that as we increase the concentration of the medication, concentration of the drug-filled nanoparticles, we see that even when pH equals 7, none of the virus particles reach the target. So this is sort of the goal, is to make sure that as each of these nanoparticles sticks to each of the virus particles that are in the region, and once they stick, the nanoparticle bursts open and the drug sort of kills the virus on the spot, and making sure that the virus doesn't reach the target. These are my references and my acknowledgements. Thank you for listening. I'm going to stop sharing my screen. Okay, how do I do that? Wait, can you make me stop sharing my screen?
Let me see.
I don't know why I cannot see.
I'm just going to share my desktop. Okay, here, let me add mine.
Yes.
Cool. Thanks, Anu. That was a great talk. Does anyone have any questions?
I'll ask a quick question. Where do you think the I guess I maybe wasn't 100% clear on it, but where the experiments data like played the biggest role in the model formulation, or like how you ran the simulations?
More than yes, in the very beginning, to get all those values like the 3.3 and the 13.9, all those values, because the information that we had from the experiments was the diffusion coefficient, and that's what we used. And they have like, I think the way they did it was they put like a small tracker on the virus, and they saw how the virus moved and how much time it took for it to sort of get the exact value of the diffusion coefficients. And yeah, that's the only data we had, and we sort of extrapolated from there.
Got it. Makes sense. Cool.
Ami, you can just unmute yourself.
Well, you know, I don't want to be official with this. So I was actually curious for the model that you created with this, like on the computation side, what is like the average runtime, and like, I guess from the beginning when you started, what was it running at, and then were you able to make it more efficient, and like what like libraries you're using and things like that?
I used no libraries. It was just MATLAB. And in the beginning, I was running into memory issues, but then I started using like decomposition and other functions from MATLAB, which sort of reduced how much time it took. Even like, because I was doing the A inverse, I sort of saved the LU decomposition and everything, just making, because that's not going to change. Initially, I was recomputing it every single time for every single time step. But yeah, just those little changes. And it takes a long time when I have it plot every single time step. But on an average, I think just for the 13.9 hours, it takes just maybe a minute or 2 minutes to compute. Yeah.
Oh, that's pretty fast. That's cool. I was actually also curious about the like grid spacing and stuff like that. They're using like step sizes for spatial and time.
I don't remember. It's been a while. I think the step sizes was 1000 by 1000 grid. So I have 10 micrometers, and then I had 10 space steps. I think for time, I just kept changing it to just see how much it matters. I think I was going to almost milliseconds as the time, each time step. But again, I don't remember the exact number. It's just, I've not touched it ever since. So
No problem. Thank you.
yes.
I have a question that's not directly with your presentation, but more of like, how have you seen your research impact your just beginning your new job? Have you seen the skills be able to roll over into your job, or are you having to learn a whole bunch of new skills?
I am learning a lot of new stuff in my job. I think I think I learned patience more than anything doing this project, because nothing worked for a really long time. And that sort of happens at work too. Nothing really works in the beginning. And I just have to ask like 20 people how to do everything. So I think that rolled over. I still feel like I don't know anything almost all the time. Yeah, but in terms of skills, I think, because end of the day, it's all programming, just knowing how to Google and knowing what to ask and just where to look for whatever you need. Yeah. But I don't do anything math related or any PD or anything at the moment.
Cool. Thank you.
Yeah.
Any more questions in the audience? No? Okay, well, with that, I'm going to thank Anu again. Thank you so much for your talk.
Thank
Congrats
you.
again on graduating and like starting your real adult life. It's unbelievable, but I'm so proud. And yeah, thank you again for giving your talk.
Thank you.
Cool. Okay, so we'll be back next week. Jared's giving a talk on ocean modeling, and I hope to see you guys all next week. Cool. Thanks, guys. I'm gonna end it here. Thank you.
Bye-bye.